So we're going to continue our conversation about action potentials. We almost finished this last time. This was the end of chapter 11. And remember we talked about action potentials are always the same. The cell, the neuron, reaches threshold, and that threshold is triggered at the axon hillock, or what's called the trigger zone. And then that signal is going to go racing down the synapse, or down the axon, and it'll go faster by saltatory conduction if there's myelin there. It'll go slower if there isn't myelin, an unmyelinated axon. We talked about the whole sodium and potassium deal that's going on. We mentioned the refractory periods, that as soon as, we're talking milliseconds, but as the sodium and potassium gates are opening and closing, there is nothing you can do to make it happen again. So there's a period of time called the absolute refractory period. During time, absolutely nothing can trigger it. And then there's a relative refractory period where it's relatively more difficult, but not impossible, to set up another action potential down the axon. So with all that behind us, this image is the chemical synapse. Where there's two synapses, two types, chemical and electrical. And I'm just going to pick up right where we were, and we'll fill in any gaps along the way. And if there are any questions, please don't hesitate to ask. The image here, we're looking at one neuron at the very, very tippy end at the axon terminus at the synaptic knob, speaking to the next neuron. And that is referred to as a synapse. The space between is called the synaptic cleft. And what we see here, remember that an action potential has come down, right? It's come down the length of the axon, and there are mitochondria down here. Okay, that's something just to keep in mind, that usually we think of mitochondria being up in the soma area, and yes, they are there for energy production, but there are some mitochondria down here at the very end of the synaptic knob, and we'll talk about how they got there later today. And then we see that when the action potentials come down, what did I tell you comes into the synaptic knob? Calcium. And what kind of gate allows that calcium in? It's a calcium channel. We would call it a voltage-gated calcium channel because it's the voltage difference when the action potential comes down that's going to kick open that calcium channel. And what does that calcium do? It binds to the vesicles that are already there, and those synaptic vesicles are already filled with a neurotransmitter. In this case, the neurotransmitter we're talking about is acetylcholine. So that acetylcholine is a protein. It's already made. It's in the vesicle. Where was that acetylcholine made? Way, way back in the soma, right? That could be in the next county over. You know, it's a long, long way away for some very long neurons. But that protein was made back in the ribosomes, in the soma, packaged in the Golgi, sent off in a vesicle, which then made its way all the way down the axon, and then stores itself here at the synaptic knob. And when the calcium comes in, it binds to the vesicle and causes an exocytotic event. And now those vesicles are going to open up, and the acetylcholine is going to be released into the synaptic cleft. It's a very short distance. That acetylcholine is going to kind of waft across the little space and be picked up on the other side, down here at the postsynaptic membrane. And there will be some channels. Those channels are going to be sodium channels. They're not going to open in response to voltage. They're going to open in response to the acetylcholine. And so we're going to call those ligand-gated sodium channels. Right, or chemical gated, but you'll hear ligand or chemical gated sodium channels. And then we can imagine what's going to happen, right? So once those, and there's the interaction shown in the cartoon, once the acetylcholine binds, it's going to open up the gate. Sodium, always found more on the outside, is going to come rushing in. And now you can imagine that we're going to set up another electrical disturbance in the next neuron. Now that electrical disturbance that we're setting up could be called an local, right, potential. It's not an action potential, right? Action potentials only are initiated at the axon hillock, at the trigger zone, or what we call sometimes the initial segment, right, where the axon attaches to the soma. So this would be another local potential, and if enough of this disturbance occurs, right, it might reach the threshold and cause another action potential down the neuron. But at this point, this would just be a local disturbance. Now, we'd be in trouble if that acetylcholine was allowed to just stay there. If the acetylcholine stayed there in the cleft, it would keep triggering the sodium gates to be open. So there has to be, and there is, a mechanism in place to quickly get rid of any acetylcholine or whatever other neurotransmitter we might be talking about later on. But in this case, there's an enzyme called acetylcholine esterase, ACHE. And it sits around in there, and what it's going to do is immediately start kind of wiping the slate clean, getting rid of all that acetylcholine, so that the next neuron is not always being excited. So it allows us to have control and to also have rapid firing, right? So we have control and rapid firing occurring because of the acetylcholine esterase that is there. Now that is a chemical synapse. That's the basic idea of a chemical synapse. And then remember too, there's the possibility of synaptic fatigue, right? That the neurotransmitters just get worn out. There's so much stimulation that there can be a delay going on. That's a possibility. And there could be delays as a result of that. Not too common, but it can occur. I just want you to be aware of it, that there can be delay. And that then the other kind of synapse is an electrical synapse. This is where we finished up. And electrical synapses that you've heard about before would be like in the heart. The intercalated discs really are an electrical synapse, and that's how the cardiac muscle, when the action potential comes down, and we'll get into the details of this in a few weeks, but when the heart, the atria and the ventricles begin to contract, all of the cells contract as a single unit, because the cells are being stimulated almost simultaneously, because the signal doesn't have to go from electrical to chemical and back to electrical. It just goes one cell to the next, right through these pores, if you will, the intercalated discs that allow the electrical signal to travel from one cell right into the next. It's nice, it's fast, but it doesn't have much control, right? So that's where we talked about at the end, that all of these chemical synapses, while they do kind of slow down the overall transmission of signals, does give the nervous system tremendous control and adaptive nature. And we'll talk more about that control as we continue this last little bit in this chapter. Here's just an image of the electrical synapse. And again, the pre and the postsynaptic cells come not just close to each other, but truly are touching each other, the membranes, and the electrical signal can travel right through a space or a gap. And that's, again, what intercalated discs are in the cardiac muscle. So, any thoughts, concerns, questions thus far? I have not yet put out a quiz yet for Chapter 11. I will now be doing that, okay? So I'll be looking for that in the next couple of days. You had the quiz on Chapter 25, and now you'll have a quiz on 11, and that will help you, again, prepare for all things related to the exam. Always think about doing the homework assignment, then the quiz, right? Or do the dynamic study modules in the homework, and then the quiz, and then you'll be ready for the exam. And those will be rolling out sequentially. I'm not making them all available to you all at once, because I think there's something to be said for doing it, progressively as we move through the content. Are there any concerns, mastering, or assignment issues? Yes?
I just have another question. Back to the chemical synapse.
Mm-hmm.
You said that it's a local.
What's happening, back to the chemical synapse?
Because in one part it says it's actually potential. And then
It has the potential of setting up another action potential
if it meets threshold.
if it meets threshold.
So how would you know that by the millivolts?
Yes, hold on to that thought. We're going to hit there right now. Okay, perfect. It's a good segue. The question was, some places it says that the sodium gates are going to set up another action potential. That is potentially true. Not always true, but potentially true. Hold on to that, and let's see where that wishy-washiness to my answer comes into. Okay? So let's take a look. I finished up saying that the postsynaptic, right, the cell that's receiving this, is going to basically get a graded potential, aka a local potential, and we can call these, in addition to calling them local potentials, we can also call them EPSPs and IPSPs. EPSP is an excitatory postsynaptic potential. The word kind of tells you what it is. It's excitatory, it's on the postsynaptic neuron, and it's changing the membrane potential. So the EPSP is moving, if you think about the voltmeter and you think about the action potential graph, an EPSP is going to be moving the curve where? Which way? Up or down? Up, right? Moving it toward threshold. Okay, so excitatory things are going to move it up, but not very much. Okay, each of these little EPSPs are little tiny disturbances. If enough of them happen, then we may reach threshold, but these are those little local potentials. So we're just calling them, we're giving them different names. If it's an excitatory potential, it's an EPSP. Okay? Now, there's also inhibitory. Now, this is something I haven't discussed yet. Well, I did say that local potentials could be positive or negative, right? I said that. But this is how they work. An IPSP would cause the cell to become hyperpolarized. Right? In other words, for the curve to go downward, making it more difficult to reach threshold. Can you imagine with me, what would that look like from an ion standpoint if there was something that caused the cell to be hyperpolarized? What's going on? If sodium rushes in, that's going to be excitatory, isn't it? If sodium rushes in, the inside's becoming more positive, that's going up, right? What sorts of things would happen, what kind of gates would open such that there would be a hyper downward polarization? Rather than a sodium gate, it could be a, I heard potassium, and then I'll get to you. Potassium gate, you agree. If the neurotransmitter causes a potassium gate to open, what does potassium want to do? Get out, right? It's always more inside than outside. So if the ligand, if the neurotransmitter, if the excitable moment caused not a sodium channel to open, but through a different chemistry, a potassium channel to open, that would cause a loss of positive charges from inside the cell, leaving the inside more negative, hyperpolarized. So potassium channels, we can appreciate that that could cause the cell to become hyperpolarized, yes? You with me? Then the other possibility, which is not uncommon either, is a chlorine channel. We haven't talked about chlorine channels, but they exist. And if the chemical caused a chlorine channel to open, where is chlorine found in greater concentration? Remember, chlorine always hangs out where sodium does, so there's always going to be more chlorine on the outside of the cell. If you opened a chlorine channel, chlorine would be flowing in, making it inside more negative, and once again causing the downward slope, a hyperpolarization. So what we're seeing is that it's not all about plus, right? It's not always about sodium, but there can be, and there are, this intricate interaction of potassium channels, chlorine channels, and sodium channels. And then what's going to happen is that you got, and I'll show you a picture in a moment to kind of get your mind around this. Textbooks always show one little synaptic knob coming in contact with the postsynaptic cell. Remember, there can be thousands. We know that in the brain, some neurons are talking to 10,000, as many as 100,000 other neurons at the same time, right? So you've got tens of thousands of signals coming in at the same time. Some of those signals could be excitatory, EPSPs. Some of those signals could be inhibitory, IPSPs, by a combination of all these ions flowing in and out. And the cell is going to add them all together. And we call this summation. So there's going to be summation of all of these signals. Now these signals, as you know, are coming in at the millisecond level, right? We're talking, things so fast, even, Michael Phelps didn't win by that little bit, right? So we're talking millisecond stuff. But if all of this stuff comes in pretty much simultaneously, the cell is going to integrate, sum up all of those signals, and basically end up with with an answer, right? And if there's more positives, then there would be a positive effect. If there are more negatives, there would be a negative effect. If there was an equal amount of both, it would just cancel each other out and there'd be no net effect. Okay, and I'll show this graphically to you in a moment. So here's the graph, and we're going to use a different word here. If there's an EPSP, we recognize that's typically a sodium channel opening. The curve's going to go up. We haven't reached threshold, but that little EPSP, we're going to call that facilitation. We are facilitating or possibly facilitating another action potential to be formed. So there's little disturbances, little EPSPs. And then there could also be little IPSPs, right, where you have inhibition. So a facilitation is going up, inhibition is going down. If we had both a facilitating and an inhibitory signal at the same time, basically, as we sum them together, it would be a null, right? Nothing really will have occurred. So now imagine this being not one or two little disturbances, but hundreds, thousands simultaneously occurring. And this is what the business of the neuron is. It is receiving all these signals in simultaneously. So this is called integration, right? We're going to integrate all of this incoming information, and again, it is phenomenal that cells can do this. Now, where is threshold always going to be? Threshold will always be met for an action potential at the axon hillock. Remember I told you too that axons are coming in, they're speaking to the dendrites of the axon, or of the neuron, and they're speaking to the soma. And those local potentials, little EPSPs and IPSPs, only will travel a short distance. So there has to be a great number of these little local EPSPs and IPSPs in great enough magnitude to cause the membrane at the synapse, or sorry, at the hillock to reach threshold and to cause the action potential. So it's all about the massive number of signals coming in at once. So again, we're just going to pull them all together, and sometimes this is more simple, sometimes there's a greater amount of control going on that I'll show you. This is a cartoon representation. Back there is a soma. Can't even see it, right? And what we're getting a sense of is that this isn't one little axon speaking to one little axon. This is hundreds and thousands all speaking simultaneously, and the soma can be just completely covered by other axon terminals, right? So this is just a tremendous kind of communication system. So if each and every, this is just a roundabout number, but each and every of those little EPSPs and IPSPs only account for a very small, maybe half of a millivolt difference. And there would be numerous, right, millivolt differences needed. What's the resting membrane potential? Negative 70. We have to get up to negative 60, and it's really kind of confusing. Hold with me there. Some things in your book are saying negative 60. Other things in your book are saying negative 55. I usually teach negative 55, but a couple of these images are saying negative 60, so that the data is a little bit in that area. But that means that we have to go from negative 70 up to negative 60, negative 55, right? That's a 10 to 15 millivolt difference. Each of these little IPSPs and EPSPs is only changing it by a half. So there has to be the summation and quite a few of them coming in to cause that action potential. There's two kinds of summation, temporal and spatial. When I say temporal, what comes to mind besides a part of the brain? Keeping the time, right? Keeping the tempo. So temporal summation we're going to see is related to timing of the signals. Spatial summation is going to be related to the three-dimensional arrangement, the spatial connection between these different signals. So let's take a look first at temporal summation. For temporal summation, we are imagining a single synapse that is being fired rapidly, one after the other. Now look at this. The typical EPSP lasts for about 20 milliseconds. So what, and I'll show you a graph in a minute, but what this means is that you've got a single neuron firing rapidly in succession down the same axon. It's tapping very quickly, a fast tempo. If that happens, that one signal repeatedly can push the threshold, push the axon to the threshold, okay? And we'll see how that can work. That's temporal. Think about it. If each time the action potential goes down to the terminus, acetylcholine is released, right? And there's a finite amount of time where the acetylcholine is in the cleft and then it has to be erased by the acetylcholine esterase. If you keep pumping action potentials and chlorine or calcium down there and you keep releasing acetylcholine, at some point the acetylcholine esterase can't clean it up and you're going to get this constant signal, and you're going to push the postsynaptic cell to threshold. and here's a picture of that, just to give you an idea of this. So temporal summation, you've got, this is two arrows coming down. It's hard to see. There's two arrows coming down the same axon terminus, right? And is stimulating the axon. And not just two, but multiple very, very rapidly. And that would cause the threshold to be released. Notice where we are. That's the axon hillock region, right? So we're gonna knock that axon over into threshold and cause the action potential to go down, right? The next neuron. Versus spatial summation. Now we're not talking about one synapse rapidly firing. Now we're talking about a collection of individual synapses all bringing in a signal at the same approximate time. So again, the neuron that's receiving all this is seeing a greater number of these excitatory EPSPs. Okay? And so that's going to, again, be summed up, summation. If they're all facilitating, it's going to cause a threshold to be reached. So now we're not talking about one signal coming down, but we're talking about signals coming down from multiple axon terminals to the same soma at the same time, many of them coming in, again, causing threshold to be reached and action potential to be generated. So two different kinds of summation. Okay. Now, are they in isolation? No. I mean, these things happen at the same time, right? So there would be temporal summation and spatial summation all being coordinated in some magnificent way on a single neuron. It's overwhelming. It's just amazing to think about how rapidly our nervous system communicates and all the magnificent control that is exhibited here. So look at EPSPs and IPSPs. Now, the last thing I want to talk about is how is all this being processed? Yes, most of this is being processed in the background, right? We're not aware of all this. This is all happening. Sometimes, though, there are going to be neurons that help to control this. I told you that because the chemicals in the synapses, although it does slow down the overall speed, it does increase the control that the nervous system has over all these signals that are running around in the cell and in the body. So we're going to talk about some regulatory neurons, okay, that are going to facilitate, increase, or inhibit, decrease the likelihood of the neuron to fire. In this image, I know yours are in black and white, the two top neurons are labeled as regulatory neurons. Okay, they just look like two other neurons, right? But notice what they're doing, especially the one here on the right. This neuron is coming down and speaking to this postsynaptic neuron, right? The green one's going to the purple. But so is the blue one, right? So we see that there's two neurons coming down there, but look where this synapse is. This synapse is where? Axon to axon. Now, I mentioned this last semester. This is an axo-axonic synapse. And we really don't think about these very much. They weren't in our thinking last semester. Last semester, I always said, you know what? The axon comes down and speaks to a dendrite, axodendritic, or the axon comes down and speaks to the soma, axosomatic synapse. I said to you there were axons that talk to axons, and that seemed really weird when I maybe described that to you last semester, and I said, well, hold on to that thought. Well, here's that time. So what do you think this green neuron is doing? This green neuron is going to somehow influence, regulate the signals that are sent by this presynaptic neuron. It could be positive, it could be negative. Right? So it's not just one cell speaking in series to another cell. You have other neurons that are coming in and influencing signals from other neurons. Again, the complexity, the levels of complexity become mind-boggling as we think about these regulatory neurons. So this neuron, right, this one could be regulating this guy, and then this one could also be regulating it. Again, just if you can imagine an arrangement, it exists within the nervous system. Now, acetylcholine is the only neurotransmitter that I've discussed. It is by far the most common neurotransmitter in your body. It is the one that controls all of your muscle movement in your body. It is the one always used as an example with the acetylcholine esterase. It's a nice example. However, there are over 100 different neurotransmitters used. So not only do you have all this complexity just with one neurotransmitter, you have hundreds or 100 plus neurotransmitters. Each one works in a slightly different way. Each one can open and close different kinds of channels, excitatory or inhibitory. They have different reaction times. Again, increasing the complexity by having these chemicals involved as intermediates to the nervous system. They all have some effect on the ion channels, okay? So, the sodium, potassium, chlorine kind of thing. And some of them work through G proteins. And I'm not going to spend time right now yet talking about G proteins. Just know it's another way that cells communicate with each other. Okay, so sometimes we have a direct access to an ion channel. Sometimes we work through a G protein channel. And I'm just not going to go there quite yet. Just know that the number of possibilities just became exponential as I said that to you. Now, I know you can't see this. You can read this in your book. Acetylcholine. Don't worry about the structures of these, please. But we've seen acetylcholine. We see that it's the one that's primarily working on our muscles. But you've all heard of norepinephrine, right? Norepinephrine and epinephrine, those also are neurotransmitters. We think of epinephrine as being a hormone-like molecule, but it also crosses over and is very much involved as a neurotransmitter. So we see norepinephrine and epinephrine. Serotonin, I think that one came along the way. We all hear about serotonin inhibitor, reuptake inhibitors, the antidepressive medications. Serotonin is an important neurotransmitter within the brain. It affects mood. Glutamate, another really important one that we'll see along the way. It's really important in memory. All of these so far are typically thought to be excitatory. They all excite. Acetylcholine excites muscle to contract. Epinephrine excites our body. Glutamate, serotonin, they all have an excitatory effect. There's one more, though, GABA, okay, G-A-B-A. This is a very important neurotransmitter within the central nervous system. And what I want you to see is that it is an inhibitory, okay? So when I say GABA, I want you to think, that is one of those inhibitory neurotransmitters. And look how it works. It tells us it opens chlorine channels. We describe what that would look like. And it can indirectly open potassium channels. Does that make sense to you? That if I open chlorine channels or potassium channels, either of those would be hyperpolarizing and basically be IPSPs. Whereas everything else here, you're going to see it's excitatory, which typically means it's going to facilitate through an opening of a sodium channel. Are those connections being made? Okay, that's a really important idea. Grab onto that, listen to me say it a couple more times on this if you need to do that. Okay, so we recognize that our nervous system is interpreting all these amazing signals. One of the things I'll be going into, we're going to be talking about special senses and touch and sight and hearing in this little nervous system unit. And it's still amazing to me to think that even though when light comes into your eye, right, it's a photoreceptor that light triggers this, but what's traveling down the neuron is still an action potential. And when sound waves come in and bend the hairs in your ear, what's triggered? An action potential. And every action potential is exactly the same way. And somehow the brain interprets all these different action potentials caused by touch, chemicals, vision, right? All those things are still communicating through the nervous system by this little flip of sodium and potassium ions. Cool stuff. So we have to figure out how to interpret this information. All these action potentials are traveling along axons, and the brain then has to figure out, okay, does that axon, boy, it came from the eye, so that must be related to vision. This axon is coming in, it was excited, it was triggered by a chemical, maybe about taste or something. And we know that these signals are going to different parts of the brain, don't we? So that's where we're kind of heading. As I move into the next chapter, we're going to be talking about different parts of the brain, some of which you know, quite a bit of which will be brand new for you. And you're going to see that where those neurons end up in the brain is kind of how the brain in part tells where it came from, right? So you know that signals that go to the occipital lobe That's primarily what kind of signals? When I say occipital lobe, you should be thinking visual, right? Remember the eye and the signals that come in for vision go mostly to the occipital lobe, right? So we're going to be talking about some of those pathways and how is it that the brain is able to interpret an action potential coming into the brain as being either a visual signal, a hearing signal, a touch signal, a smell signal. Now, this goes back to, well, not goes back to, this is back to our acetylcholine story, and we'll talk about this again when we get to muscles in a couple of weeks. But as you can see here, this is the frequency of action potentials. Think summation. If the neuron is traveling with 20 action potentials per second, that's amazing. 20 action potentials, 20 signals are zinging down an axon 20 times a second. That is slow enough, honestly, for the muscles to contract and then relax, contract and relax, contract and relax, contract and relax. And notice every time the green arrow goes down to baseline. We're going to call that a twitch. Your muscles can twitch, right? Very, very fast. We all can experience this faster than what we can even imagine. If the signals are coming down at around 50 times per second, whoa, I cannot rest in between that. So I will have a little bit of a twitch and a little bit of a twitch, but notice how this is going up. There's never enough time in between the stimuli for the acetylcholine to be erased and for the effect to be brought back down to the resting. And then, if the signal's coming 100 or so times per second, I will reach what's called tetanus. Now, when you hear the word tetanus, you usually think of a shot, right? But tetanus is a disease, a condition caused by bacteria, which causes your muscles to go into a spaz. Okay, and we'll talk more about that later. But what happens is that the muscles are in a complete spaz. They're completely contracting. There's absolutely no relaxation. Boy, you would never want that to happen to your heart, would you? If your heart muscle spat and was in a total tetanus, it wouldn't be contracting, right? It would never stop contracting. It wouldn't relax, and it wouldn't be able to pump blood. So we'll talk when we get to the heart, how the heart never goes into tetanus, and there's special reasons why that is. But your other muscles can go into tetanus. If you had a cramp, you know that your muscles went into a tetanus situation, right? They got very, very stiff, and you weren't able to relax them. So again, this all comes down to the number of action potentials going down to a muscle 20 times a second, no problem. The muscle can actually relax if it's coming 20 times per second. So that comes back to the idea of summation. And then you can read about this whole idea about processing and facilitation and all those good things. So that brings us to the end of chapter 11. The big story here is action potentials and the interaction and the different ways that action potentials can interact with other neurons. You should definitely be able to, with great confidence, draw out this curve going back to, you know where I'm going. Slide 57. No doubt about it. You should be able to draw that out freehand and show me the resting membrane potential, show me the threshold, show me the maximum plus 30, and label the depolarizing, repolarizing, and hyperpolarizing events, okay, on the curve. No doubt about it. So you should expect right now on the test to have a little short answer thing and a little box that says draw out the typical action potential and label it appropriately. Okay, so no doubt about it, that needs to be something you can close your eyes and draw out with great confidence. That's probably the biggest, that's the one thing you need to be able to do physically. The rest of the test, at least for this chapter, would be multiple choice, but that would be something that might be freehand, draw out, short answer kind of thing. Okay. Questions, thoughts, concerns on chapter 11, or even anything back to chapter 25? Now do you see why we did 25 first, right? Sodium, potassium. Now we got sodium, potassium down. Now we're going to keep moving along with some brain stuff. Yes, sir?
For the action potential, do you want us to know like, when all the channels are opening and closing and stuff like that?
Yeah, so you should also be able to appreciate what's going on, right? So at threshold, what happens? First of all, where does threshold happen? At the
Axon hillock.
axon hillock. And one of those videos, and I said too, what is it about the axon hillock? There's a huge number of those voltage-gated sodium channels at the axon hillock, such that they all go flying open, and that's what causes that rapid depolarizing event. And then what happened at the top of the curve? All those sodium channels closed, and potassium channels were just a little bit slower to open up, and now they open up, and that brings it back down, and they always overshoot hyperpolarizing before we come back to threshold. Other thoughts? Who's feeling like, I've got this? I just need to look over, but I've got it. Few confidence. Who's like, I have no idea what you've been saying for the last 2.5 hours? Okay. I mean, it's been a while, right? And and the rest of you were somewhere in between. Okay. So and there weren't too many on the extremes, right? So a lot of you in the kind of, I don't know what he's talking about stuff. What are you going to do? Take a look at some of the homework problems. They'll attack this from different ways. And then, if you haven't, and I mentioned to some who were earlier, I found a few extra copies of the worksheets. And these were just copies that Pearson sent me last summer. And if you don't have one of these, take one of these. But there's some really nice, these are the same as the chapter review things at the end of your modules in your book. But you can scribble on these. They're also available through Mastering. You can find a link to some of these. But there's the pieces of the chemical synapse, if you wanted to label that. There are parts of a neuron. So there's some nice little activities in here. And I'm not going to, quote, assign these and say turn them in, but this might be a good place to go, right? So just as a way to break it down. And the good news is all the answers are in the back. So there's never going to be any wonder if you're labeling something incorrectly. So that's a tool for you in addition to everything in Mastering. And then remember that in Mastering, if you go to Chapter 11, it's going to have pre-tests and post-tests and activities and chapter guides and more than there's time in the day to do. So again, find 2 or 3 of those tools and grab them, do them for 25, 11, 13, and see how the test goes with that preparation. And then if that's not enough, we'll make some adjustments. And also, I've sent out reminders about the resource room, right? We've got tutors who are available for one-on-ones. We've got walk-in tutoring with Janae and Andrew. They've posted their schedule outside the door. I'll try to remember to post it. Andrew, who's a well-experienced tutor, he's in the nursing program, and his schedule's moving around a little bit, so he's going to be kind of posting his hours a couple weeks at a time. But there's still quite a few hours of walk-in tutoring available to you, in addition to my office hours and Mr. Mueller, whoever else that you can grab and ask about these good things. Yes, sir?
Okay, so we're not doing all 25 on the test, just like the 20.
Just what I talked about,
Just what you talked
right?
about.
And you'll see that at the beginning, just before you go on, you'll see that at the beginning of each of these chapters, as I move over to our segue to Chapter 13, I'm going to say, and it's on your packet as well, which of the modules that I'm focusing on here as a presentation and the modules that you want to focus on, and then it's telling you which ones to review. Now, I'm not going to test you directly on, quote, the review modules. That's the stuff that was essentially covered in 105. But would some of that 105 material inform your understanding of this material? Absolutely. So you may want to at least flip through the pretty pictures in the Martini book, make sure you know what I think you should know, not to spend hours on it, but you may find that, if you've forgotten where the occipital lobe is, that it's going to make a difference to you as we move forward. So focus on, if I have not, as I'm putting together the exam, another way of saying it, as I'm putting together the exam, I'm not going to test you on things that I have not specifically discussed. So if you come across a concept or an idea in the Martini book or in the study books or in the study modules or anything like that that I have not touched on, just let it go. Right? Don't stress about it. Oh, am I supposed to know that? I don't remember that. If I talked about it, yeah, I want you to know about it. If I didn't talk about it, you can just kind of like, okay, go on. Some of these assignments, and I've said this, I think, some of these assignments are kind of prefab. Some of them I have control over. Some of them are a little bit more modules, and I have to assign the entire module to you, and I may not have discussed a small portion of those. And you're going to get to that in the homework and go, oh, shoot, is that something I'm supposed to have understood? If you get to one of those things, it's like, man, this is nothing he talked about. Click on the first answer you see and move on and don't worry about it.
Modules,
Let me look at that. Let me look. I thought they were going to be on schedule, but I may have been off by a day. Let me look at that, Tom. Thank you. Yes, sir. Ma'am.
Mine are also making me do them correct, like get the correct answer, or I have to keep repeating it over. Like
That's the idea.
But I mean, if there's this stuff that you didn't teach.
That can be the frustrating. There shouldn't be
It
too
won't
many
let
of those
you
things.
continue until-
Yeah, it won't let you continue to answer it right? So get it right the first time or make a note of it. Okay, the right answer for that crazy answer is A, and just don't forget it. I know, that can be a little frustrating, and that's the price that has to be paid because the 80%, 90% of those things are really, really good. Okay. Any other complaints, green apple or green tomatoes? Any other things to sling at me right now? That's okay, I'm not taking it bad. Will I put the answers up for the ones you got wrong for the quiz? Yes. Did that not show up yet? So I should be able to, I'll remind myself to do that for the Chapter 25 quiz, and I'll make sure that those answers get up for you. Okay, so you can see what you might have missed. Okay, that's probably a setting on my part. It would not become available until everyone had taken it, but I should be able to go back in now and set it so that you can see your responses. Yes, ma'am. Is it tonight? Yeah, so it's possible that it's going to be showing after midnight because that's when it's due, the last chance, right? So I'll look at the settings and make sure it's possible, but that's why. Even if I had done it right, and I think I did, you wouldn't be able to see it until after until the deadline is passed for everybody. Okay, moving on. Chapter 13. This is the chapter about the brain and about cranial nerves and about pathways. And you are to review some of the more 105 type topics from the first 3, and then 5, 10 through 11. Those are going to be brain anatomy and structures that should be familiar to you. Now, a couple really, really quick things here to get our minds reminded about the significant structures of the brain, and then I'll be adding to this, to your understanding. So remember, the brain has on its surface a bunch of gyri, right? All these gyruses, and they are separated by little grooves called sulci. And we have the cerebrum, which would be what we consider the lobes portion of the brain. And how many lobes are there altogether? Altogether on both sides? 10. Woohoo! What are we forgetting about? 2 frontals, 2 temporals, 2 occipitals, 2 parietals, and we're forgetting about the 2 insulas, right? That hidden insulated lobe deep inside the brain. So there's actually 10 lobes, 5 on each side, and those lobes have particular jobs that they're assigned to, and they also are talking to the other sections of the brain. And then remember, there's a cerebellum. And then there's a spinal cord, and in this area right through here where I'm circling, that would be called the brain stem areas, and I'm going to highlight that for you in a moment. So here's that brain stem, kind of on a stick, okay, brain stem on a stick. And what you're seeing here is the diencephalon. You remember what that word means? The diencephalon was the pit, the core, the center of the brain. It's not officially a part of the brain of the cerebrum. The cerebrum are the lobes, right? The fleshy part of the peach, the fleshy part of the brain, and the diencephalon, the pit, the core, the very center. The part of the diencephalon includes the thalamus, and you may recall I always said the thalamus is the gateway to the cerebrum, so everything coming up the spinal cord, going through the brain stem, is going to pass through the thalamus on the way into our consciousness, into its way into the cerebrum. Then there is the hypothalamus, which is not showing here, but if you can imagine the sagittal section of the brain, remember the hypothalamus was always kind of shown under the thalamus, as the name suggests, right? Hypo, under the thalamus, and you'll see another view of that in a moment. Then there's the brain stem, and the areas of the brain stem include a midbrain. Now that's just this little area right here, difficult to point to on most models, as well as the pons and the medulla oblongata. Okay, so the medulla and the pons and this little area called the midbrain are part of the brain stem. Remember, too, that the brain has some fluid-filled regions within it called ventricles, and there is 2 lateral ventricles, left and right. So we've got two lateral ventricles. Those are shown here, right, on the left and the right side. And then there's a third ventricle. That third ventricle is in and around the diencephalon. So right here where the thalamus is, there's a fluid-filled chamber around there, the third ventricle. The fourth ventricle is down deeper in the brainstem, down between basically the pons and the cerebellum. And then those ventricles and the fluid within those ventricles is continuous with the fluid of the cerebral spinal fluid that is going up and down the central canal of the spinal cord, as well as the fluid that is circulating around your brain that your brain is sitting in buoyancy in. Okay, so that's that CSF, that cerebral spinal fluid. Looking at it from a different view, okay, so now around the outside of the brain, what's that stuff called? The outer layer of the brain is called the cortex, right, the cortex. And all the inner layer, the deeper stuff is the medulla or the medullary area. We can see the blue-colored ventricles. We can see the cerebellum, okay. What lobe, just to get you thinking, what lobe is right here where I'm circling? What's the lobe we forgot about? The insula. And how would I find the insula? If I had a brain in my hand, how would you find the insula? You would lift up, right? Lift up where? On the frontal parietal area and pull down on the temporal, right? Along that lateral sulcus, and peeking back there would be another whole set of gyri and another little lobe, okay? That would be the insula. Again, I'm not going to go back and quiz you directly on this anatomy, the basic lobes, but I will be discussing the functional areas of the brain and what each part of the brain is responsible for. So you do want to get this back in your head. If I slice the brain sagittally, remember we've got this big old band, we've got this big old band of white tissue, right? That band of white matter is the corpus callosum. And then pretty much just underneath it, this area is the diencephalon, including the thalamus. And in this image, I can see that little triangular region. That is the hypothalamus. And then hanging from the hypothalamus, it's not shown here, but there would be hanging off a little stalk, the pituitary gland. I can see the cerebellum. And the pineal gland, we'll be talking about the pineal gland, is that little tiny gland right here. It's all labeled for you. But the pineal gland is in the shape of a pine cone, and that's why it's back there. And it's also part of the diencephalon. Now, I bring you back to this part of the brain. There's another part of the brain called the limbic system. If you've taken a psych course or two, maybe you've heard about the limbic system. It's not like a body system like the cardiovascular system or the digestive system, but it sounds like it might be. And the limbic system is this really cool part of your brain that includes, and let's remind ourselves of some vocabulary, some tracts and nuclei. Ooh, that's a review, right? What in the world is a tract? It's a set of nerves found in the central nervous system, right? We don't have nerves inside the spinal cord. We don't have nerves inside the brain. Instead, we call them tracts. And a nucleus, nuclei, remember this? A nucleus is a cluster of cell bodies found in the central nervous system. So we're in the brain, right? So we're talking about the central nervous system. So we're not using the words ganglia and nerves. We're using the terms tracts and nuclei. The limbic system is a cluster of structures around the diencephalon, around that pit, that core, that center of the brain. And other books would refer to the limbic system as the motivational system, or the part of the brain that is motivating. It's going to be very much involved with your emotional states. It's going to be the linking area between your consciousness in the cerebrum and your unconsciousness in the brain stem. So it's kind of a connector between the two. And it's also going to be a big part of your memory and your ability to retrieve information, and it's going to affect your motivation. Okay, so that's the limbic system, all those really cool things, you know, that psychologists talk about, behavior and modification. That's all focused here in this area called the limbic system. Now, what are the parts? The parts of the limbic system include, and I'm talking about, remember this is the link between the cerebrum and the diencephalon, so there's parts in both. In the cerebrum, we've got the cingulate gyrus, and I'll show this to you in a moment. It's a gyrus, right? A noodle, right? It's one of the noodles. So it's a gyrus. There is the parahippocampal gyrus, another gyrus, another noodle, and the hippocampus itself. So those are the three parts that are actually up in the cerebral region. And then in the diencephalon, in that pit, that core region, there is a thalamic nucleus, two of them, nuclei. When you think of thalamus, there's really two. There's left and right half, right? So there's two cerebella, right, left and right. There's two of these. When I think about the diencephalon and I say the thalamus, keep in mind there's two of these units, right, within there, left and right. So that's why it's nuclei, plural. The hypothalamus and the mammillary body that I'll describe to you. So those are the pieces, the parts of the limbic system. And they're right there at the interface between the consciousness and the unconsciousness, the cerebrum and the brain stem regions. So let's take a look. And this is going to take, you know, probably more than just these little tiny pictures to learn this. So you're going to be grabbing your book and looking at Area 13 or Module 13 and taking a look at this. So let's take a look. We've got, do we all find where we are here? Let's start in the very center. That is the thalamus. Okay. And the white band is what? We know that. We know the thalamus and we know the corpus callosum. The cingulate gyrus is the big green noodle. See the big green noodle? Right? So it's the superior, right? The cingulate gyrus is this bump. It's the superior portion of the limbic zone or the limbic lobe. And then there's the parahippocampus gyrus. That is this lower, okay? So we've got these two bumps, these two noodles, two gyri that are bordering, right, the limbic region, cingulate gyrus and the parahippocampus. What's para mean? Next to, near, right? So the parahippocampus must be near the hippocampus, and that red area, this red, represents the hippocampus. So these are portions of the limbic anatomy, the limbic structures. That would be the portions that are from the cerebrum, right? They're outside the nugget. Inside the nugget, inside the diencephalon, here we're going to have a thalamic nucleus. It's this orange area right here. Now, what again is a nucleus? A cluster of cell bodies found within the central nervous system. What color would cell bodies be? White or gray? Axons are white because they're coated with myelin made by what cells? Oligodendrocytes, right? We're in the central nervous system, so the axons would be white because of the myelin made by oligodendrocytes. The cell bodies would be gray. So a nucleus, a cluster of cell bodies, would be gray in color. So these nuclei will be little gray clusters within the white matter of the brain. Then the hypothalamus itself, we can recognize that, and the mammillary body, tiny, I know, this little pink dude, right there. Okay? So your job, learn the anatomy of those 6 or 7 things, the cerebral part and the diencephalon part of the limbic system. So when I refer to them, you know what I'm talking about. Now, each of those little areas has a special job in this connection and control of your emotions. So the anterior group, that thalamic nucleus or nuclei, they're going to be the relay station. And if you look where that nucleus is, it's going to be the connector between the mammillary body and the cingulate gyrus. There's also in here the amygdala. Okay, the amygdala or the amygdaloid body. Amygdaloid is a word from the Greek meaning almond. So it's an almond-shaped area. I just call it the amygdala. And the amygdala is going to be sort of the interface. It's going to be the connector between the limbic system and your cerebrum and your sensory systems. This is going to be part of that fight or flight, and this is also going to link your emotions to specific memories. Okay? We all know that in our well-being, there are certain memories that bring to the surface emotions. Those memories, those histories, those emotions are stored in this amygdala region. The amygdala is also a region of fear. Okay, and we'll come back to that, fear and anger. The hippocampus, okay, it comes from the Greek meaning seahorse. In some angles, the hippocampus sort of has like a seahorse shape to it. Hippo is the Greek for horse, okay? And there's some very important short and long-term memory retrieval parts to the hippocampus. Now keep in mind, as I discuss functions of these regions, it also suggests that if there was damage to any of these regions, that an individual with damage to these specific little portions of the brain would have difficulty or impossible outcomes dealing with memory or anger or emotional stress or control. So damage to the limbic system can make a person rather interesting. Okay. There is a little arch area, the fornix, and we'll see that this is a little section of white matter, and it's going to connect the hippocampus with the hypothalamus. So when we look at this thing, let's think about this again. This is a blow-up. The green was the upper superior gyrus. That was the cingulate gyrus. The lower gyrus was the parahippocampal gyrus. The red center was the hippocampus. Doesn't look like much of a seahorse here, I know, but in a different direction it kind of looks like a seahorse kind of thing. The mammillary body is this little section right here, this little red guy. And the nucleus, nuclei I should say, of the hypothalamus. And look what's sticking out here, just to give you your orientation. What's sticking out right here? It says it's the olfactory tract. At the end of that olfactory tract is your olfactory nerve. And remember, that is cranial nerve number 1, related to smell. So it's kind of moving up under and is going the most anterior of all of your nerves as it comes up toward the front of the brain, comes down through your nasal passages. This purple guy, almond-shaped in some angles, is the amygdala. Okay? And then the fornix is this yellow area. That's the yellow, and that's the connector between the hippocampus and the corpus callosum. So lots of little detail. This is all brand new for most of you, right? You've not seen this little detail story about this portion of the brain. Again, I'm going to talk to you about what each of these things is doing. So, one way to approach this, when we get to this, it might be flashcards. Maybe that's the best way to do it. Just what's the part, what's it do? What's the part, what's it do? Get these terms to have meaning to you. It's pure memory. I know it's pure memorization. These things don't all make sense, but you'll learn hippocampus, memory. You'll learn amygdala, anger. Right? So just get these associations going so that you can pick out key ideas. If you're presented with a patient who's had damage to a particular area of the brain, you would be able to predict their unusuals, right? What were their idiosyncrasies based upon, what were their deficits based upon damage to certain parts of the brain. Okay? That's where we're kind of going. Hypothalamic nuclei, again, where it was. And again, this has a lot to do with fear and rage and pain and some of those really visceral reactions that occur. This is also an area, though, of pleasure or sexual arousal. So areas in here can be both pleasant, if you will, as well as fearful. And some of these areas are kind of vague. They really kind of are. Now, we're not quite done with the anatomy yet. There's also an area of the brain referred to as the basal nuclei, or you'll hear the basal nucleus. And this is the bottom, right, basal side of the cerebrum. And again, it's a nucleus. So that's a what? A cluster of gray matter, a cluster of cell bodies. And we're going to see that it is deep to the lateral ventricles. And here we're going to have some unconscious, subconscious control of skeletal muscle. We're kind of moving up. When we think of skeletal muscle, we think of voluntary, right? And we're kind of moving out of the true brainstem, subconscious, no control areas of the brain. And the basal nucleus is kind of in that intermediate area. Damage to this part of the brain you've heard of, because this is where Parkinson's issues occur. They're in the basal nucleus. So we're beginning to see that there's a connection between this part of the brain and voluntary movement. And people with Parkinsonian tremors have these involuntary tremors, right, of their skeletal muscles. So individuals who have problems in this area, or as we'll talk about in, there's a problem with some of the neurotransmitters being released from this area, they're going to have issues with initiating and having normal patterns, and they're going to have problems sometimes with coordinating things they already know how to do. So they know how to brush their teeth, they know how to comb their hair, they know how to pour coffee, but there's an issue with those memory, muscle connections with a person with Parkinson's because there's an issue in this part of the brain. Now there's so you've got 6 or 7 parts of the limbic system to learn from an anatomical standpoint. You've got a few parts also of the basal nuclei. It's plural, nuclei. Turns out that there's 3 major parts here. There is the caudate nucleus, the globus pallidus, and the putamen. So I'll show these to you in a picture in a moment. So there's 3 areas, the caudate nucleus, the globus pallidus, and the putamen. And this is all in an area called the internal capsule. Let's take a look. So we've got a sort of an odd view here. We're looking at a couple different levels. They're both transverse, but at different levels, so you can see this. And I'll show you another picture in a moment. The caudate nucleus, okay, is the inner one. Then within the internal capsule, you've got the putamen, this one, and you see the thalamus also shown here. And you can also get a glimpse of the pineal gland sitting here in the back. So this is kind of an odd view. I wouldn't ask you to label this particular view. And there's the fornix as well, that yellow area that's connecting everything. I would instead want you to know this view, right? So put a little star on this one. This one, I think, lays things out in a more appreciable way. This is the frontal section, right? Cutting the face off, frontal or coronal section. And what do we call this groove right here? That deep groove right there is the... Nope, this is this way. So that thing is the longitudinal fissure, right? That long groove that separates the left from the right hemisphere, right? Now you with me? And then in this view, the corpus callosum would be this area right through here, connecting the left and the right, okay? So here are those nuclei. The caudate nucleus, where is it? It's right up underneath the ventricle. And then there's the putamen and the globus pallidus. So 3 little nuclei, 3 little islands of cell bodies amongst all the white matter of the medullary regions of the brain. And then just to give you context as well, the amygdala, we already talked about the amygdaloid or the amygdala, that's the purple. Okay, so you've got on both sides, right? You've got these nuclei. We're in, again, that sort of in that connection area between the unconscious and the conscious basal nuclei. So what's going on here? What is the basal nucleus doing? Well, it's going to help modify movements. As I already mentioned, problems in this area, like Parkinson's, are going to give you overstimulation and issues with control of your movement. And there's also a connection in this area between output to the cortex after going through the thalamus. So you could, one of the things this does is it helps to anticipate movement. You may not even think about it because these things happen so fast, but even before you voluntarily move a skeletal muscle, your subconscious and part of your basal nucleus has already started adjusting the muscle tone in anticipation of your voluntary movement, right? So these are all very subtle differences, but problems here would have issues with coordinated movement. And here's just a chart, and just to give you the idea here. So when you think of the motor area of your cerebral cortex, right? That is what? That is cerebral cortex. We're talking about the voluntary regions of your brain. I'm choosing to do this. And when you think about it, there's a direct connection. I choose to do it, therefore I'm going to tell my muscles to voluntarily move, right? I think it happens. That's the direct connection that we think about for the motor side. And that's true. But on the sensory side, you can see that the basal nucleus, the caudate, the putamen, the globus pallidus are also speaking to, indirectly, to your motor areas. So they are what? Modifying, influencing, right? They're not necessarily in control of, but they're certainly influencing your ability to voluntarily move your skeletal muscles. And you also see that it influences the motor areas, but there's also the ability of the basal nucleus to kind of almost directly control your movements. This would be subconscious, or this would be when there's an issue with this part of the brain, as in Parkinson's. So you are not choosing to have tremors, right? But the basal nucleus, because of its issues, is directly sending signals, causing your muscles to move uncontrollably in those tremor-like motions. Everyone know what I mean by Parkinsonian tremors? Right? Is there anybody who doesn't know? Individuals start having just uncontrolled tremors, and they become rather substantial, and they really become debilitating at the time. Okay, so that's just kind of giving you an idea of how this whole thing works together. So again, what does the basal nucleus do? What do the different things do? And if you had damage to the basal nucleus, what would you expect? Again, control, inability to control your muscular movement. Again, think Parkinson's disease. Okay, so that's a little bit of anatomy, right, that we've never talked about, but we need to understand those new anatomical regions to appreciate the complexity of the brain. And I know I'm kind of gotten away from it, but remember, all these parts of your brain are talking to each other through action potentials, right? So we don't want to lose track that that is the way by which all these portions of the brain are going to be talking to each other through these electrical things called action potentials. Thoughts, questions, concerns about limbic or basal nucleus? Limbic system or basal nucleus? When I say hippocampus, you're thinking memory. When I say amygdala, you're thinking
Anger.
fear, anger. When I say limbic system, your overall well-being, emotional status, well-being things, right? And when I say basal nucleus, there's three parts to it, but they're all really doing what? Helping to control movement or coordinate the movement along with your cerebral, your voluntary control. Okay. Remember, too, about the pre- and post-central gyrus? You remember this story? That we're up in the cerebrum. So in the cerebrum, I heard someone say central sulcus earlier. What's the central sulcus? The divider between the frontal lobe and the parietal lobe. And on each side of the central sulcus, remember there is a very important noodle, right? A gyrus. And in the frontal lobe, you have what's referred to as the precentral gyrus. You also would call that the motor strip. Remember that? The motor strip and the frontal lobe being all involved with motor control. And in the parietal lobe, there is the postcentral gyrus, which many people refer to as the sensory strip. So that strip of brain matter is controlling your senses, right, and receiving your sensory information. Then remember all the other lobes. We mentioned in passing we had the frontal lobe, parietal lobe, occipital lobe, temporal lobe. What we can't see here, insula lobe. And how would I find it? I would take a fork and I would go right in this area and I would lift up on the frontal and pull down on the temporal, and right back there would be hiding the insula lobe. I guess 2 forks, right? Okay, so what I'm going to do now is probably go into an area that's more familiar to you. We're heading up into the functional regions of the cerebrum. So when I say visual cortex, remember the cortex is the outer layer of your brain. So there is a portion of your brain in the outer layer called the visual cortex, and the visual cortex is back where? On the occipital lobe. Okay, but I'm going to show you a couple more names for this. We're definitely back on the occipital lobe. But the visual cortex is going to be interpreting and understanding all things related to seeing and visual things. Oh, there's my forks, right? I see my 2 forks? So if I pull back on this area, what's pointing back there is the insula. And this picture shows us some of these primary association areas. So again, visual area back on the occipital lobe. See it pointing there. So there's your primary visual cortex back here in the occipital lobe. There are primary sensory areas. Well, when I say sensory, what lobe comes to mind? Where is the sensory strip? Parietal lobe, right? So the sensory strip and the primary sensory areas of your brain are in the parietal lobe area. When I say motor cortex, control of movement voluntarily is primarily up in the frontal lobe, at the motor strip. There's also gustatory cortex. That's the part of the brain that's receiving what sort of stimuli? Taste. It's back on the insula. Okay, so taste is back on the insula, as is the olfactory. So the smell and the taste are kind of hidden back there, much on the insula. Auditory cortex, your hearing, is primarily your temporal lobe area. That makes sense, near the ears. Okay, so the auditory area on the temporal lobe. Okay, so again, those are the areas I want you thinking about: vision, occipital, sensory, parietal, motor, frontal, auditory, temporal. Because we got one major job for each of the major lobes, and then taste and smell kind of hidden back a little bit more close to the insula. So know those, and obviously these things are integrated together. There are these integrative centers that don't mean, when you think about language, right? Language is not just seeing. Language is processing, sensory. It is seeing, visual. It is hearing, right? All of those things have to come into the brain. And even though I've just suggested to you that there's certain pockets of the brain that do just one thing, it's not quite that simple. All of those signals are also going to be integrated together. Okay, and one of the big things, speech, writing. Writing is what? It's motor, right? It's also spatial. It's visual. Mathematics. Okay, I mean that's a foreign language for a lot of us, right? So there's a lot of areas of our understanding and our everyday doing that don't just go to one part of the brain, but are integrated by multiple areas. So we're just going to call this integrative centers, and some of these integrative centers are actually focused on one side of the brain or the other. And this is perhaps something you've heard of, and that is that your left and your right brain are not identical. They're not just mere images of each other, although most textbooks would suggest that at first glance. When you look more closely, you'll see that there are some things that occur on one side or the other in most individuals. And for example, the speech center, also called the Broca's area. This is something my students have heard about before. The Broca's area, also called the motor speech area. This is an area that is usually on the same side of your brain as the general interpretive area. Hold on to that. And this is going to be, not only, this is motor speech. So this is your ability to articulate, to breathe, and to move your muscles of your tongue and your throat and your pharynx, and all that's necessary to speak is coordinated on just one side of the brain. Okay, so you have a Broca's area. And if you had damage to that part of your brain, you would not be able to effectively speak. And you may know of individuals who've had a stroke or a head injury, and after which their ability to understand is just fine. But they just can't get the words out. They've lost that. So that if you were to go back and look at an MRI of their damage of the brain where there was what part of the brain was damaged by the stroke, you would see clearly that Broca's area had in some way been affected by that brain damage. Okay, so that's an example of one of these integrative centers. Another integrative center is in the prefrontal cortex. The prefrontal cortex is where you are going to be performing abstract intellectual things. This is where your intentions come from. This is in the frontal lobe. And this part of the brain is one of the last parts of the brain to fully myelinate and to fully form. Yeah, it's all there in an 18-year-old, but it really doesn't completely finish its final architecture of myelination and processing until about age 25. And you'll hear people say, oh, their prefrontal cortex, they're not thinking very well. That's an excuse of the young. But also damage to this part of the brain would leave people unable to think abstractly, maybe to have inappropriate actions toward others, maybe to have kind of weird personalities. So a lot of that can occur if there's damage to the prefrontal cortex. There's also the frontal eye field. This is an area that controls your learned eye movement. So reading and moving your eye in predictable ways, that's only on one side of your brain than the other. And then there's this thing called the general interpretive area. Another name for this is Wernicke's area. That you've heard from me before. It's another area. This is more processing of sensory information. So Broca's was about speaking, motor speech, coordination. Wernicke's area is more about understanding what's being said to you and the processing of incoming information. This is usually also only on one hemisphere, typically on the left side. So a person who has damage to the left side of their brain, would potentially have damage to Wernicke's area. Now there are people for whom their brain is completely like a mirror image. So most people have the left side have Wernicke's, but there are people who have Wernicke's on the right side. And they walk around, they're normal people, but needless to say, the brain is not always arranged in the way that we may describe it to you. There are exceptions to the rule. So this area is going to be integrating all of that incoming sensory information. So where are these? Let's take a look. Motor speech, Broca's area. Motor speech. What lobe are we in? That yellow is where? Here's my central canal. Central. So that's frontal lobe, right? And when you think of frontal, you think motor. And this is motor speech. So this is the coordination of all things related to movement of tongue and mouth. This is your general interpretive area, Wernicke's. Okay? So damage there. Again, what side of the brain is this? This is left side. Frontal eye field, right? Eye movements, trained eye movements. Again, frontal lobe, right? And we're actually where? Let me erase this. Watch this. What's this strip of land right here? Motor strip. Pre-central gyrus or motor strip. Doesn't it make sense that the eye movement would be, on the motor strip? Okay, it's in that area of the frontal lobe. And then all this blue area up here, this is that prefrontal cortex. This is the very front of the brain, the most anterior portions. And that's where you're thinking about inhibition areas and executive thinking and processing of some things. And like I said, that's the area that's last to develop in your brain. So this brings up the idea that your two hemispheres, left and right, are not identical, And we refer to this as lateralization, right? There are certain things that occur on most people on the left side. There are certain parts of the brain that are only found on the right side. The left side is pretty much where your speech centers are, right? Broca's and Wernicke's on most people, left side. It's also where you have your general interpretive area. That was at Wernicke's area, right? All that's on the left side. And this is where most of your language-based skills are found. And for most people, right, the premotor cortex is larger on the left side for right-handed people and vice versa. And that whole part of the brain is important for analytical tasks. So people, your mathematical abilities, your logical centers are more on this side of the brain, on the left side, versus the right hemisphere. The right hemisphere is more of an abstract side. It's picking up more sensory information, but it's really sort of your ability to analyze emotional things is over on the right side. So a person who had damage or had deficits on the right side may have a difficult time understanding these two phrases. You know, get lost, get lost. They just can't interpret the meaning of something. So again, they're missing some of that interpretive ability. This is also side of the brain that's involved with 3D relationships, artistic sorts of things, psychological, not all, but many psychiatrists, people who are psychologists, counselors, are artists, musicians, will tend to be right brain dominant, and they are therefore left-handed, right? So people who are left-handed are right brain dominant, and if you look at the general population of artists and musicians and psychologists and architects, there's a larger, higher percentage of those individuals who do have a left-handed nature. Okay, so it's not an all or none, but it definitely there's data that suggests that's a pretty strong correlation. So here's a picture of these two sides. So again, the left side where I started, speech centers, right? The writing centers, that Wernicke's area, that general interpretive area. And then on the other side, you've got your touchy, visual analysis, shapes, artistic kinds of things going on. Remember, you still have the corpus callosum. It's connecting the two sides. So these two sides are not in isolation. They are talking to each other, but your two halves are not mirror images, right? And neurologists and neuroscientists will talk about the right brain and the left brain, just like cardiovascular people will talk about the right heart and the left heart. So they are matched up, they're side by side, they're working together, but they have some individuality to the pair. So left-handedness, here we've got a stat about 9%, 10%, right, of the population, 1 in 11, something like that, are left-handed, and their motor cortices, right, they flip over just like the rest of us. And again, here it mentions it in print that a higher percentage of those musicians and artistic kind of folks are left-handed because of this lateralization. Okay, I've only got a couple minutes. That's like the little brain anatomy thing that I wanted to share with you. The last thing in this chapter that I want to get to, and I'm clearly not going to finish it today, but I just want to introduce it, are EEGs. Okay? All of you have heard about EKGs or ECGs. That's electrical activity of the heart. And maybe you've heard about EMGs, looking at the electrical activity of muscle. And EEGs, electroencephalograms, are measuring the electrical activity of the brain. And so we can put electrodes on the head and measure these electrical activities just like we can EKG from the chest or EMG having electrodes somewhere on the musculature. And the brain is sending out predictable brain waves. These are the same brain waves that are used to determine if someone is, quote, brain dead. If no longer brain waves are being generated, then the person is declared brain dead, and depending upon the situation, right, they might be considered brain dead or dead for the purpose of transplantation of organs, that kind of thing. So we're going to be looking at brain waves through an EEG, looking at brain waves. And again, memory, All right, this is a memorization kind of thing. There's some different waves that are coming off the brain. They're just alpha, beta kind of terms. So the alpha waves are going to be in a normal, healthy, awake individual. They're resting with their eyes closed. Okay? We don't have alpha waves when we're sleeping. We only have alpha waves when you're awake, eyes closed, healthy, awake. Okay? And they also disappear when you're concentrating. So if you're really, maybe right now you have lots of alpha waves because you're really not with me, or maybe you have no alpha waves because you're really thinking about this and you're really focusing on something. If you were doing a lot of mathematical calculations, your alpha waves would go away. Okay. Beta waves, beta waves are higher frequency, they occur more often, and they're going to appear again when you're concentrating. So if you're concentrating, your alphas go away, your betas come in. Stress, maybe you're feeling stressed. When you come in for a test, you have a lot of beta waves going on. And any kind of psychological tension, anything that where the brain is feeling stressed, psychologically you're stressed, your amygdala is turned on, lots of beta waves. Theta waves, okay? Theta waves are going to appear transiently during sleep. They also will be more observable in frustrated adults, and if they're there, often it may indicate some sort of brain disorder. So there's something going on that's not normal. These guys are a little bit more intermittent. Then delta waves are really large amplitude, big high waves, but low frequency. They don't occur very often, and they're going to be seen when you're sleeping. Okay, so these are your sleeping waves, and you'll also see these if there's problems with the brain. So an adult with a tumor or there's a blood flow issue to the brain, or some other part of the brain, then you would inappropriately see delta waves in a normal awake individual. So we can use these different waves. I'm not going to ask you to recognize them on an EEG, but you can hook up electrodes to the brain, see these different waves, and see them at predictable times in a normal individual, and you would see them spike in abnormal situations. We'll finish up with sleep and what happens to these waves during sleep. I hope you get some sleep. Right, over the next few days. That's a good segue. And I'm really not that far from finishing 13. If on the way in you did not get chapter 15, I'll start that next time. Go ahead and have it for your packet. And those who show up next time will get 15. If you're listening to me and you never come to lecture, 15 is posted on Blackboard for you. And if you want to grab a copy of the worksheets, if you don't have one of those, please take one of those on the way out.